Good afternoon, ladies and gentlemen, and a warm welcome to a new Wetsus webinar. My name is Michele Tedesco. I'm Scientific Project Manager at Wetsus, and today I will be your host. The topic for today's webinar is the future of carbon valorization. It's a huge topic, as you all know, and today we will try to cover some of the latest developments in terms of electrochemical and biological conversion of CO2 to valuable products, and looking specifically at what could be the role of water technology in this field. We will do this with four speakers, our guest speakers today. The first is online from Aarhus University, Dr. Uffe Sognstrup Thomsen. Can you hear me, Uffe? Welcome.
Yes, please.
From Aarhus University and the Novo Nordisk Foundation CO2 Research Center. Welcome. Dr. Tom Sleutels from Wetsus and University of Groningen. Dr. Legrand from Coval Energy, and Dr. Nico Claessens from Groningen University and Research. Thank you all for being here. So before we start, some small information about how we'll work with the questions. So you can ask questions in the chat. There will be a QR code for that should be soon on the screen. So basically, if you want to ask chat to the speakers, just scan the QR code now, and the QR code will also be repeated after every presentation, so you can ask questions in the chat. We will moderate the question here in the room, and we will try, of course, to answer as many as possible in the time limit that we have for today. So now I would like to give the floor to the first speaker. So Dr. Uffe Sognstrup Thomsen. Dr. Thomsen has a PhD in marine biochemistry and 20 years of experience as a consultant in the water and oil industry. He's been working since 2020 at Aarhus University as a consultant for water engineer scientist, and specifically looking at collaborations between universities and industries. So in his role, he is actively involved in CO2 capture and green transition. Today, he will give us a more broad perspective about the green transition and also the opportunities in water research. So please, Uffe, the floor is yours.
Thank you. I'll just remind you that there's a lot of echo. I hope it's only me, but there's a lot of echo when you speak. Yes, I will give you a brief introduction to what we do here in Aarhus University on the energy side, CO2, and also a little bit about the water science that we do, because I know that Wetsus is also interested in that part. My talk here today will be a brief overview of Aarhus University and who we are and what we do. I will tell you a little bit about WATEC, the interdisciplinary center that we have at Aarhus University, then the CORC CO2 Research Center, and then I will give you a presentation for Dr. Marta Victoria. She's an associate professor at Department of Mechanical and Production Engineering, with the title, Which Handles Do We Need to Tweak to Speed Up the Green Transition in Europe? So she could not be here today, so I will be standing in for her and giving a more generic talk of our research. This is Aarhus University. It's actually a quite beautiful university. It was built in 1928, so quite old, and it's in the city center of Aarhus. So the yellow buildings are the older ones, and if you can see my pointer, we have enlarged it with the red areas over here, and near the harbor, we have a building down here. My institute, which is biological and chemical engineering, is in the buildings here. And then outside the picture, in the lower part, we have a lot of digital science going on also. So it's quite large university, one of the biggest ones here in Denmark. We have different locations in Denmark. I I hope you can see my pointer, but Aarhus here is in Jutland. And I would like to highlight this site called Viborg Forum. This is the site where we have agriculture science, but also a lot of the science around the energy side, like Power to X and stuff like that. Actually, very nearby, there is a data center from Apple who have built that center because we have so stable energy supply of green energy. So that's one of the reasons why there is focus on Denmark because of this green energy supply. I can also mention that we have big plans about windmill farms in the east around the island called Bornholm. And then on the west coast, we have plans of really large windmill farms, which will supplying not only Denmark, but also the whole region here with green energy coupled to Power to X so that we can make synthetic fuels and also hydrogen. Then I would like to mention that we of course also have an oil production like you have in the Netherlands. It's in the North Sea here. And there's also a transition going on there. There's 2 projects going on with CO2 storage. One with Total Energies, where Shell is partner, and one with Ineos, that you probably also know, a British Switzerland company. So these companies are actually also involved in the green transition here in Denmark. But WATEC is an interdisciplinary center, and this is something we have at the university because we want to promote interdisciplinarity at the university. So there's 5, you can say, clusters, and I'm heading the one called pipe water. So all the water that runs in pipes, wastewater treatment plants, and so on. Then we have a group with sensors. They work on sensors for the water industry, and also a lot of for the actual research part where they supply very, very small glass sensors for oxygen and so on. Then we have natural waters, which is fresh water. We have a land water interface and instruments. That's a lot about agriculture. And then, of course, economy and management is also important because what is really the cost benefit for society. Then I would like to highlight that we every year in August have a summer school called Advanced Water Management course. And if you're interested to join or have students that would like to join, you're very welcome. We have a website called watec.au.dk where you can look into that. Then there's also a center called Digitus, which is all about digitalization. I'm sorry about the noise. I'm in a hall here. So the pipe water cluster, we work with these topics: drinking water supply, wastewater treatment, resources and energy, and digitalization. Here I can highlight the resources and energy. This is a pilot plant where we use hydrothermal liquefaction. That means that we, from organic waste sources, can produce biocrude. So that's just an example of what we have ongoing of research. Then we have the CO2 center, which is quite new. It's almost started, and that's one of the reasons why I am sitting here today, because they are overwhelmed by people who want to invite them into webinars. So that's why I am standing here today. I can hear mention that 15th of February there is a research talk where you can are welcome to join. It's with Professor Matteo Pascali from Rice University in US, and it with Professor Alfred Sporman. He's actually the director of the center. There's huge interest in this center, so if you have any questions or something you want to follow up, I think it's better that you reach out to either Stephanie Christiansen or Mads Bendiksen, or actually me. I have a lot about the water science to do. So that's a little bit about what we have ongoing. I can also maybe mention that alone in the Water Center, we have 100 researchers from PhD to full professors. So I will start up with the slides from my colleague Marta Victoria, and you're also welcome to reach out to her if you have very specific questions. It's a bit top-down approach to what the topics are today, but I think it's important because it's gives you some answers to why are we actually doing this this green transition. This is a graph from 2019, the gas demand, and I guess you all know the story about Ukraine and so on. But the large gas demand in Europe is in Germany, it's in UK, and it's in Italy. But actually, I can see that the Netherlands also are really dependent on gas supplies. And from the data you can see in 2019, a large fraction of that gas came from Russia and from Norway. If I should guess what the data would look like today, Russia is close to zero, and we have much more from the Middle East, like Qatar. You can also see in the figure, in the graph, in the low right corner, that a lot of the gas is actually used for heating residentials, and then there's a lot of gas demand in the industry and for also power plants. Yes, I would like to this is the paper that Marta Victoria was part of, long-term implication of reduced gas imports on the decarbonization of the European energy system. And here are the take-home messages. The 1.5 degree path quickly release Europe's dependency on imported gas. We have a massive ramp up of wind and solar photovoltaic capacities required for the 1.5 degree. Limiting gas requires ramping up heat pumps, and then we need to employ the biogas potential fully utilized. This cost optimal systems takes in these equations. It's generation, it's storage, and it's transmission cost, and then it's of course variable costs. And these are subject to generation plus balance equals demand. And then we have a constraint with the CO2 emissions with a CO2 limit. So cost and technology parameters assumption for the DEA technology data catalog. This is what she includes in her model. This is a picture of the sector coupled model of the European energy system. It's comprising electricity, heating, transport, industry, and feedstock and carbon cycle. And here you can see the electricity demand in Europe. Not surprisingly, it's the large economies like France, Germany, UK, and Italy that has the largest demand of electricity and energy as a whole. To the right, you can see this industry activity coupled to the emissions, and you can see essentially you need to couple the demand with the distribution network, and this is the challenging part. And the more wind and solar energy we will include, the more fluctuations we will see, and that's why a really high resolution model in time and space is needed. This graph, I guess you're also familiar with. This is cumulative CO2 emissions since 1850, and then you have the temperature raise of the global temperature. And you can see that if we should aim for the 1.5 objective, then we only have 400 gigatons of CO2. But if we aim for the 2-degree objective, then we have more CO2 emissions allowed. So the path for the 1.5 degree objective would be much faster lower of CO2 emissions, and we need to start up really soon. I can tell you that in Denmark, the government objective is to decrease CO2 emissions by 70% by 2030. So we are already on track on this. But if we take the 2-degree objective, then it's slower, and then we need to be much faster in the end as we approach 2050. The 1.5 degree path quickly relieves Europe's dependency of imported gas. So this has actually forced us to do something. And you can see if we take the 1.5 degree objective, then gas, coal, and lignite needs to be removed really fast by 2025. But if we take the 2-degree objective, then it's a slower decrease in these fossil fuels. Yes. So a massive ramp up of wind and solar photovoltaic capacities is required for the 1.5 degree objective. So you can see that already in the year 2025, we need to raise those capacities significantly. But if you take the 2-degree objective, it's a slower and more straight increase. Then, if we look at the heat pumps, limiting gas requires ramping up heat pumps much faster. And as you can see, over a year, there's a high demand of heating in the colder months, of course, and so the heating is really the variable part that requires electricity. The REP Power EU targets 10 million heat pumps by 2025, but the model here shows that actually 20 million is needed. Biogas potentially fully utilized. The potential assumed for biogas in Europe is 320 terawatt hour per year, and this is fully utilized by 2030 in the 1.5 degree scenario, and by 2050 in the 2-degree scenario. A similar amount of 400 terawatt hour per year of synthetic gas is used by 2050 in both scenarios. This I can tell you, and maybe just a side remark here is that in Denmark we have a big biogas production. It's called Nature Energy, the company, and I know that actually Shell had just bought that company. So we have a good relationship with the Netherlands. So summing up, the 1.5 degree path quickly relieves Europe's dependency on imported gas. Massive ramp up for wind and solar photovoltaic capacities is required for the 1.5 degree. Limiting gas requires ramping up heat pump faster, and then biogas potential fully utilized. I think that was my final slide. I hope I'm on time, so I can stand here for some questions.
Yes, you are on time. Thank you very much also for keeping your time and for your presentation. So I see there are at the moment no question in the chat, so maybe I can ask myself with some questions. So thanks for this general overview of nice also to see this sector coupled model with all the different infrastructure. What I perhaps miss, and it's a hot topic in the Netherlands, is the role of hydrogen. Can you elaborate maybe a little bit on that? What's the role of hydrogen in relationship to power to X, which is also the topic for today?
Yes, I can at least tell you what's ongoing in Denmark is that we are in the process of building these windmill farms, and this requires that we need to transfer the electricity to something else. And here, power to X plants are actually going to be built both in the west and in the east, so they will transfer electricity into hydrogen in the first place. So, and that also requires a network of pipes for hydrogen, and that's also in the planning in Denmark at the moment. And the next thing is that we need to use that hydrogen for something, and that's also a power to X technology where we will couple CO2 from biogas production to hydrogen to produce these synthetic fuels. And that's also something that we are planning to do in collaboration with Maersk, which are our big company for container shipping.
All right, thank you very much. I think in view of the time, I think we need to stop here. So I would like to thank you again.
Yes, and maybe I can say as a last thing, use me as a contact point for if you have any question. I work with both energy and the water science, so I think it would be good to reach out to me if you have further things you want to discuss.
Definitely, we will stay in touch, and I will share also your presentation with the participants after this talk. So thanks again.
Thank you for having me.
Thank you. Our next speaker is Dr. Tom Sleutels. Sitting next to him, a colleague of mine of the Wetsus. So Tom has a background in biotechnology and a PhD in environmental technology from Groningen University. He now combines his time as a scientific project manager at Wetsus and as a scientific staff member at the University of Groningen. So please, Tom, the floor is yours.
Yes, thank you very much, Michele. It's a pleasure to give a talk here about phototrophic biomass production. That will be the topic of today. So we're talking about microalgae. Microalgae use CO2 as a carbon source. So I think it's a very suitable topic. And they use light energy as the sole energy source for growth. Now, many people have for decades been pursuing the growth of these microorganisms for very interesting products. And that is because microalgae are actually a very wide range of organisms. So they are able to produce a very wide range of interesting components. And within microalgae themselves, they also consist of several components that could be interesting for production. So they have been pursued for the production of biofuels, for example, chemicals. You can use them as food or feed, maybe. They have been produced for food additives and other applications. Now, the downside of growing phototrophic biomass is actually that it's not as simple as it seems. So you have a wide range of microorganisms that are capable of producing these things, but they also all have their optimum of growth. So they need a special light intensity or color of light to produce certain compounds. You need to produce them in a clean environment to be able to have a clean production system. Actually, they also produce oxygen in the process of photosynthesis, which is limiting their growth. Now, you also have some limitations in the production systems that they are growing in. And most of these limitations are area related. So you need to harvest sunlight to be able to grow these microorganisms. That means that you need a very large area for cultivation systems. On top of that, if you grow a lot of biomass, the biomass itself starts shielding the rest of the biomass for the light that they need. So producing large quantities of this biomass is quite difficult. So all in all, the production of biomass is quite expensive, and that is why they're only applied for relatively expensive products at the moment. So it would be great that if you could increase the thickness, so the density of this biomass production, that it also comes available for the production of other compounds. Now, if you look at what factors could be improved to increase the biomass productivity, actually the photosynthetic efficiency, so the efficiency in which they use the light to harvest energy to grow, is the most important parameter. And also the light path. So here's an example on top of the reduction from 2 to 1 centimeter has a huge impact in the reduction of the cost. Of course, this also comes at the expense of producing less biomass. So this is not what you want to do. But I want you to keep in mind that the efficiency of light and the area that is available are very important parameters. Now, we have been looking at these organisms, and we have thought of a method to actually increase the growth. And our approach is based on feeding them with hydrogen as an additional energy source. So, as I said, they usually use light to fixate CO2, but most of these microorganisms are also able to actually produce hydrogen under special conditions. So they produce enzymes called hydrogenases that are able to convert protons into hydrogen. And many of these enzymes are reversible, so they can do the opposite reaction. So we've been thinking that if you feed them hydrogen, this could serve as an additional energy source for growth. Feeding them hydrogen as a gas could be fed all over the reactor and should have access to all the biomass that you're growing. And in that sense, it doesn't have an area limitation. So usually you have photosynthesis, you have two main photosystems that harvest the light. You have photosystem 2 and photosystem 1. They are used, the energy of the light is used in these complexes to oxidize hydrogen to electrons and protons. These flow through a wide range of complexes in the cell membrane, and finally these electrons end up in the reduction of NADP to NADPH, and the protons are used to form ATP. And these are the general energy carriers in microorganisms that are used to build biomass. Now we know from several microorganisms that the hydrogenases that are present in these systems are actually connected to photosystem 2. So in theory, the oxidation of hydrogen to protons could inject additional electrons into these cells that is then available as energy for additional growth. So overall, you have then still the process of photosynthesis happening. So in the process of photosynthesis, generally you need 8 to 10 quanta, which is packages of light, to fixate one CO2. And in photoreduction, part of this energy is donated by hydrogen, and this would require only half the amount of light to fixate the same amount of CO2. So overall, in principle, you should be able to produce twice the amount of biomass using the same amount of light energy. Of course, we wanted to test this principle. So we did some tests in a confined environment. We used closed bottles because we wanted to have them exposed to an atmosphere of hydrogen. So we had cultures growing with hydrogen in the headspace. We had cultures growing without hydrogen. So we added nitrogen gas to have the same pressure. And we did this under different light intensities. And we wanted to study if the addition of hydrogen indeed had a positive effect on the growth. So here's an example of growth of Synechocystis. So that we use that as a model organism. It's very well known. The genetic information is well known. And we know from this genetic information that the organism contains hydrogenases. So that's suitable in principle to use hydrogen as an electron donor. This graph shows the increase in biomass expressed as dry weight in time. And we exposed them to a low light intensity. And here we didn't observe any light, which is a bit peculiar because at this light intensity they should grow. But as you saw in the previous slide, the bottle contained a cap, so the light intensity probably in the bottle was a bit lower than was expressed here, but we were not able to measure it inside the bottle back then. But we were still confident that it could work, because at 100 micromoles per meter square per second, so that's an increased light intensity, we did observe growth. So the conditions were right for the biomass to growth. And then we exposed the same flask, so the same organism, to a lower light intensity, but with hydrogen in the headspace. And there we saw a quick rise in the biomass concentration, and we saw a slightly increased productivity of the biomass. So the slope of the line in its steepest point is a bit steeper than for the 100 moles. So at a lower light intensity, we were able to produce more biomass than at a higher light intensity. So for this was quite a nice finding. Of course, we analyzed the samples a bit further. So in this graph, I show the consumed hydrogen on the left side. These are the bars together with the increase in biomass, and you see that the decrease in biomass in the headspace, which we sampled, it nicely correlates with the hydrogen that was taken out of the bottle. Of course, this is not a direct proof, but it's a nice indication that indeed the hydrogen was responsible for the increase in growth. Now, we were also curious if these microorganisms could perform photoreduction without light. So here is a comparison of a culture growing in light, so continuous light, which is also the data that you saw before, which is the darker dots, with a culture that was grown first in light, and after 5 days we switched the light off, and at that point we didn't see an increase in biomass anymore, and also not a consumption of hydrogen anymore. So indeed, this theory that probably the hydrogenases are connected to the photosystems, and that they are only conductive and only utilizing the electrons from hydrogen at light is quite likely. Now, why is this an interesting option? Because hydrogen is becoming more and more popular, also in the field of reducing CO2. But if you look at algae growth, it's area dependent. So you could also put a PV system on land and look at the efficiency. So if you have the efficiency of a PV and an electrolyzer to produce the hydrogen, this is around 20%. And the efficiency of photosystem is around 2 to 6%. Now, there's many attempts to increase this efficiency, but so far it has not been very successful. So if you could achieve an increase in productivity using hydrogen, this, if you look at the area comparison, it's very interesting. Another option is that it's very likely that oxygen is used to remove the electrons that are additionally produced to produce biomass. So this could actually reduce the amount of oxygen that is available in your system. Now, oxygen is toxic for the photosystems, also for the hydrogenases, by the way. So reducing the oxygen level in your system could make it actually produce even more biomass. And the hydrogen can be supplied throughout the entire reaction, which makes it less area dependent. So that's also a very nice feature. So after this proof of principle, we are pursuing this research further in a PhD topic. So we will still use these batch bottles to study what happens to the cultures a bit more in detail. We want to study actually what happens in the pathway, what happens at different light intensities, what happens to the productivity and insight to the biochemical pathways. But we also want to move it towards a practical application. So we quickly want to optimize the system towards a more continuous system, because this is how it is applied in practice. It also allows a bit more to control the conditions. In batch, the conditions are continuously changing, while in a continuous reactor you have controlled settings. So there you can really optimize the conditions per microorganism to have optimal growth. We also want to test what happens to the content of the microorganisms. I can imagine that the pigments are maybe a bit less, but maybe other compounds are expressed more. So how this composition changes is interesting. And of course, we also want to see if this is possible in other organisms. With this knowledge, we want to go to a bit bigger scale and also to perform a techno-economical analysis. Hydrogen is for now quite expensive. The production systems for algae are also quite expensive. So if it leads to growth, is it sufficient to allow for the addition of hydrogen? That's what we want to do at the end of this project. So this is what I wanted to share with you today. If you have questions, please contact me. Type them in the chat. And
we show the QR code again. Thanks. So yeah, thank you very much, Tom. The QR code will be visible immediately on screen. So if you have more questions, you can just use the QR code to ask a question in the chat. Well, in the meantime, I do have some questions, Tom.
Nice.
So I can start. So it's a nice concept. So to basically use hydrogen as a energy carrier and reduce the light intensity, but then you need first to dissolve hydrogen in water, which does not solve that well. So do you see any problem there?
I do see a challenge there. As you mentioned, hydrogen doesn't dissolve well in water. But hydrogen is used in quite some biotechnological applications. So microorganisms are known to use hydrogen. And the trick usually is to have a high utilization rate. So if you have a low concentration of hydrogen in the liquid itself, so if it's used very fast, then the gradient for more hydrogen to dissolve will also be higher. Of course, then you still don't have a lot of hydrogen available. So this research is also sponsored by a company, and they are specialized in actually the gas exchange in phototrophic bioreactors. And I think their knowledge in helping us getting the hydrogen in the system, and also the mixture of hydrogen and oxygen actually will greatly help in moving the project forward.
Okay, yeah, clear, thanks. And what about the light? Is it about using artificial light or real light?
So in many locations in the world, there is not so much light available. So there they automatically switch to artificial light, and also because electricity is becoming cheaper and cheaper. But it has an additional advantage. If you use artificial light, then actually you can get more controlled conditions. You can also change the color of the light. And they found that this is quite determining in what the composition of your biomass will be. So if you want to steer it in a certain condition, if you want to have a certain product, then artificial light is much easier to control your product.
Okay, and maybe one last question is about indeed the products. You have mentioned there are a number of products that be made from this algae bed. Can you mention a few examples? How do you see as the most promising products?
The most promising. So nowadays, mostly related to the price, they are sold as food additives. So they're relatively expensive. And the cheaper bulk components, they will only be available on the market when the price drops. So I do see it as a food additive, for example, if you can get a nice protein composition. Some algae they produce a nice mixture of unsaturated fatty acids. So then it's still a food additive with a relatively high value. So that's fish oil basically. I think that will be a way to go for microalgae production if they become cheaper.
Yes. Definitely, that's the real challenge, right?
Yeah.
Okay, thank you very much, Tom.
You're welcome.
So I think for the time being, I would like to move on to the next speaker, which is Dr. Louis Legrand. So Louis has a PhD in environmental technology from Groningen University. He has expertise in electrochemical CO2 technologies, and since 2 years is the research manager at Coval Energy, where he focuses on electrochemical CO2 conversion to formate. So, Louis, thank you for being here with us today, and the floor is yours.
Thank you, Michele. So today I will give you a talk about or present who is Coval Energy, what we do, our goal more or less, and actually basically our approach towards valorization of CO2. So maybe quickly about who we are. So we are not a university, we are a startup company. We've been founded in 2015. We are pretty small now. We get 6 to 7 employees, 7 at the moment. We're located in Breda, and the main focus on the company is always to create value from CO2, but also to push towards commercial scale. That's really what we focus on. And recently we have been quite busy to develop our electrolyzer technology to produce formate, as Michele just said. And we do that through actually a European project in collaboration with other partners, universities and other companies, like the Consensus project and Fresh project. I will talk about it, the activities a bit later. I think a big milestone we reached this year and we continue to work on is to really build a pilot. So to go to commercial scale, if we have no choice, we have to really go to pilot, see how the system works, see the bottleneck. And that's a big, it's a big step for us, and it's the direction we want to go. So this represents a little bit about our focus. So you see a blue box with Colyzer writing on it. So that's Colyzer represents the technology we want to develop. So it's an electrochemical process. And the main concept is to take CO2, renewable power, so electricity, and water together to form chemicals, organic chemicals, like in that case, formate. And this formate itself has a limited market at the moment for now, but it can be further developed or transformed into different products, like through a fermentation process to make fatty acid, for instance, proteins, or it can be used through a fuel cell to electricity storage. And two projects we actually involved now. One of these projects is really more focused on upscaling the technology. So this electrochemical process, it's called Consensus, and the one I said is Fresh. It's a project which is more try to look at formate as an energy carrier for electricity storage. So this slide represents a little bit of philosophy. So as I said, you see now the electrolyzer, the CO2 electrolyzer, where CO2 and renewable energy comes in. And at the end, what we always want, so what Tom said, like we want to go for higher carbon product with higher value. That's what we want to do. We always want to take unwanted CO2 chemicals into valuable product. That's always a goal. But the way we want to do it, we don't really want to go in one step from electrolyzer to higher carbons like ethylene or biokerosene, a lot of products you can think about. But we first want to go take from CO2 in organic carbon, make it first an organic carbon C1, so that's a formate product. And then later on turn this formate into formic acid and use other process to make biochemical. That's what Coval Energy is actually trying to focus on. So because we want to do that, we try to partner with people who have the know-how, because if I go back, we do not know how to make chemical higher carbon compound. It's not the focus on Coval, but we do have partners who can help us to do that. And actually, my colleague created another bigger consortium. It's called GAFT. It's focusing on sustainable aviation fuel, and that's a big market who's developing right now, because for certain technology, you can use batteries or you can use hydrogen, but for plane doesn't really suit well because batteries will be too heavy or hydrogen take too much space. So we're still looking for carbon-based fuel, and that's why we think there's a big market as an end product actually for us, or a product outlet for formate. All right, so this is a little bit about how technology works out. So it's the basics of it. So it's an electrochemical reactor composed of an anode and a cathode, and some kind of things in the middle. The reaction of interest always happens on the cathode for us. So you get on the cathode part CO2 and protons together will form your formic acid. Formic acid or formate depending on the pH. So in that case, because we use bicarbonate as a catalyte, you will turn to have formate, potassium formate. But there is more things to this, to the electrochemical reactor. You also need an anode. So on the anode, we just use oxygen evolution reaction. It's kind of basic reactions that it's easiest to use. And at the moment we use a BPM, so a special membrane. It's quite useful because what we find out is BPM make our process pretty stable because it can balance the pH quite well. It's very important for the stability of the system. It can avoid crossover of product from one compartment to the other. And that's very important for us because as university, we always want to develop technology for better performance, and it's extremely good, of course, but we want also stable performance. We want to get performance that can last for a long time. It's extremely important to commercialize. So the big thing we learn from the coming years is like BPM is a good choice to go forward. And there's another particular thing about our technology. We operate at high pressure, so at 40 bars, and that's pretty straightforward because a lot of this technology are based on gas. Don't like to put CO2 and water together, but use CO2 directly, and there's a lot of benefits for that. CO2 gas is more, it's more available, I would say, in a gas form. But we don't do that because using a gas in a system always causes some problems in terms of stability. That's what we find out. So we rather take the CO2 inside a liquid phase. That works better, I think, to make the system more stable. But to compensate for the lack of availability of CO2, we increase the pressure of the system to give more availability of CO2, so to improve the system. And that you can see now kind of what it means. So this is two graphs we made with TU Delft 2019, a couple of years ago. Important graph, important study for us. Like with higher pressure, these two graphs show on the x-axis the pressure. And you get two actually key performance indicators, which is Faraday efficiency and current density. And by increasing the pressure, we can both increase the current density, which represents production rate, to make it simple, but also Faraday efficiency, which represents indirectly the energy efficiency. So that was a great way forward for us. So that means like our activities in the company when we do our research. So now you see a picture of our setup. Very simple picture. You see in the middle this big electrochemical high pressure reactor in stainless steel, because it's 40 bars, so we have to design it in a way that it's leak free and it can be safe. That's what we do. You can see the CO2 and catalyte buffer tank. So it's a small stainless steel vessel where CO2 and the KOH mix together. This is the catalyte we use to, it's how we feed the CO2 into our system. And to complete the system, you will need also an analyte buffer tank, and you need some, we call dosing pump, just to feed some fresh solution both on the analyte and catalyte. So now I can talk a little bit about the two projects we are in and more or less what's the objective. So in the consensus project, so it's a project we do with Wetsus actually, together. The goal here is to increase the TRL, so the technology readiness level of the technology, reach 6 to 7. So for us, it's more focusing on engineering. We have to upscale, and that's what you see on the picture in the middle one. We have a container actually in Breda. We have building inside. So now we finish building. We're going to commission very soon. And here, like the objective of that project and the container we're building is really to test operation time. See you can operate for 1,000 hours. See the performance. Is it stable? And we're going to do that in 3 testing locations, in Greece, Romania, and Denmark. To give a little bit more maybe context to the project, so the project has 3 parts. Capture CO2 first. So we get CO2 from a source, we will capture it. Then the CO2 have to be regenerated. The solvent to use CO2 have to be regenerated. So that's the Wetsus part. So if you want more information, please contact Wetsus, I would say. Very nice. And we are focusing on what you can see on the conversion part. So that's converting the CO2 into formic. So in that project, we're working on that. We built it. Everything went fine. But we did learn a lot. And what we learned is balance of plant is very, very important. What we realized recently that is the cost of equipment actually cost, of course. The cost is one thing. So higher pressure is nice, but there's an increase of cost. And this kind of cost, they're not very, they're quite expensive on a low scale. Like on economy of scale, high pressure equipment is cheaper when you reach a certain scale, but on lower scale, that was quite expensive. In general, there's increase of cost of materials for construction, but also for membranes, for catalysts. So it seems like the last 2 or 3 years, this cost increased quite a lot. So that's something very important when we want to commercialize later. That into consideration. And of course, what we learn a lot, and this is why it's an important milestone for us, is all know-how, how to automate this kind of plant and how to control the product quality. All right, so the second project we are involved in is a fresh project. It's a different project. This one focuses more on research and to just show a proof of principle that formic can be used as an energy carrier. So on this graph, you can see the coalizer. And on the green box, that's what Coval is focused on. So take CO2 again, energy, water to produce potassium formate. But in that project, actually, the formate is stored first and then used in a fuel cell to regenerate electricity. So it's a kind of a battery system using formate as energy carrier. And here the goal is really to show that this can be done on TRL number 4 around, but also you can do it with, of course, high KPI, so reaching quite high current density, 200 milliamps per meter square, but also high Faraday efficiency. So what it means for us, actually, we are not alone in that project, but we want to take this opportunity to develop actually kind of a new cell architecture. Zero gap, which is pretty well used around for hydrogen, for instance, but now for CO2 also, of course, it's used a lot. But as I said before, for us, the system needs to be stable. We always have stability in mind, and that's always challenging. So that's where we are going. Develop this new cell architecture, keeping stability in mind, extremely important. And all this work will not be on the large scale, but on a smaller scale, like in the high pressure setup we have. You can see on the picture that I showed a little bit earlier. This first project is also a little bit about how to operate this kind of system. So here you have an overview around about how it could look like. So that's ongoing work, having between a CO2 reduction cell, but also the direct fuel format fuel cell, to think about how to optimize both systems to increase the round trip efficiency of the overall system. So we have a lot of work to do with our partners here to improve the overall concept of the format battery. All right, so if I have to say for us, the direction seems important to us is first optimize the cost of material. And we have two materials in mind, like iridium and bipolar membranes. They are very interesting, very stable, but they increase the price a lot. So that really think about like on cost effectiveness is really the right way to go forward. We want to optimize the cost of the balance of plants, extremely important. So things we would like to do is a single pass-through. A lot of people want to do go a similar way. But also a lot of work we want to go forward now is to prioritize cell designs, as I said, zero gap, for instance, which are stable, but also could generate a lower balance of plant-related cost. If we could keep a couple of tanks, a couple of pumps, it could actually always be a decrease of cost. That's something we're looking forward to optimize. We want to optimize the cell architecture. That's another thing, like I mentioned before. And something we do with partners around is to develop market opportunities by using, finding outlet for the formate of formic acid products. Maybe take on messages you could take with you. So we at Coval Energy are doing our first pilot, so it's an important milestone, as I said, so the consensus project. A lot of our research now is on the cell architecture, new cell architecture, but towards higher current density is very important, but also good performance stability. So kind of very key here. Cost reduction in general. Balance of plant is one thing. The reactor is also another thing. So the whole overall system needs to be cheaper. Not only the reactor itself, but the whole system, whole process. And something we are always looking forward is partnership to speed up development. Always need a lot of people. You need people developing materials, in designs, finding format outlet partners. So we are always open to discuss with people and see business opportunities. All right, so thank you for your attention. So you can see all these guys here are from our team. So you have Rin, Frank, and Robert, which are the founders of the company. All have different, little bit different role in the company. So Rin and Frank are more on the business development. Robert, Declare, more on technology development. Myself, Marriott, and Jos and Marvin, we are more like the team, the employees who work actually on the daily basis in the company. So I would say thank you for your attention again. And if you have any question about it, you can always contact me. I have my email address there, or you can check our website. What I could say, if students are looking around, if students are watching now, we are always interested to work with students from different backgrounds. So if you are interested into our concept, if you want to work with us, please contact me.
Thank you very much, Louis. So the QR code will be visible online soon. If someone has questions, there is some questions from the room already. Tom,
I want to ask a question because you operate the system at quite high current densities. So what do you do to keep the anode reaction under control and not produce oxygen there and not any nasty side reactions that are harmful? You use ultra pure water, or what do you do there?
No, we don't use ultra pure water. We use normal deionized water. So now actually we were in depth in the past. Now we are in Breda, so we use even water we do from RO, like we just bought a small RO unit to make water. So going deionized water, or the specific name, I think osmotic water or whatever that is. Not the ultra pure water.
Mm-hmm. But you don't have chlorine production or anything?
No. No, I can't smell it, definitely. So no, we don't have that. It's working quite well. We use 1 molar KOH on the anolyte. Maybe that's...
There is no chloride in the system, right? You don't use chloride in the system. That's why you use only potassium carbonate hydroxide as background. And I also have some questions, Louis. So very interesting overview, by the way. Nice to see also much development in such a short amount of time that you are doing in Coval. So if you think about the reactor component itself, so what is the biggest bottleneck now that you can see, and where is also the main cost saving? You can imagine.
Like the anode, it's very expensive. If you use iridium, like we used it in the past, it was always the best stable solutions. But now we are thinking this is not the way to go forward. Apparently iridium increased a lot in cost recently. It became much less available. So going towards like alternative, that's a big one. And surprisingly, I think the BPM was increased a lot of price recently. So I'm also thinking about the relevance of using it. Cost, always from a cost perspective, it's very good for the system, but cost-wise, maybe not the best option after all. So you have to always think about the cost. Cost is always key thing.
So you might rethink the cell architecture also for upscaling.
Cell architecture is always the gas bubble, always limit the performance. So this will be an optimization step, but once again, in our approach, we really want to go for simplest design to a bit more complex, but we don't want to be too complex because stability is always an important thing for us. So like we will go step by step to complexify for the benefits of performance, but we don't want to lose stability's performance. So that's how we proceed actually. It's our philosophy.
Clear. Understand that. And in terms of main applications, you have mentioned proteins, fatty acids, electricity storage. So there are a number of ideas in terms of business opportunity. But which one is the most promising? How do you see that?
But no, actually, the format itself is limited in terms there is not so much of a market right now for it. This company has showed GAFT, so it's a venture basically between 2 companies with Coval. For plain biofuel, that's actually quite a big market there. So we're quite excited about this. And there is also a lot of different opportunities around it. Always with bioreactor, actually. It's kind of interesting, using format as a... I'm looking forward to talk of my neighbors here.
Exactly. You might find your first customer in the next speaker. But before we go to the next speaker, so then you're saying, so feedstock for bio-based product, bio-based fuels, jet fuels in particular, this is where you see the most potential for the format market.
At the moment, yes.
Okay, thank you very much.
Thank you.
Well, then let's move on to your first customer, maybe. Or anyway, the next speaker today is Dr. Nico Claessens. Nico is assistant professor in microbial synthetic metabolism at Groningen University. His research focus is engineering synthetic pathways in bacteria to support efficient use of sustainable feedstock. It can be CO2 or C1 substrates like formate or methanol, for example. So the floor is yours, Nico.
Thank you. Thanks for the nice introduction. I think it very nicely links actually to both previous talks, so I'm looking forward to follow up on these. So we look into the biology side of things. So where do we mainly cover the chemistry side? We are biologists, microbiologists. So just a bit the concept, but I will go a bit quicker to that because also Tom introduced that already. Well, so far a lot of bioproduction, so where biotechnological production of carbon-based chemicals is done, is using plant biomass, so which is dependent on photosynthesis, which is converted by microorganisms into fuels, chemicals, food, feed, etc. So, for example, the production of bioethanol by yeast from sugarcane is a famous example, but it's actually also notorious for being very inefficient because of the different steps, but especially also of biological photosynthesis not being that efficient. And the same holds, but to a lesser extent, for the production using phototrophic microorganisms. So these are, for example, cyanobacteria Tom talked about. They have the limitations in photosynthetic conversion efficiency, or microalgae. And so the concept that we work on a lot in our group is combining, and this looks directly a bit more complicated, but this will get clearer throughout my talk, is combining electrochemistry, where we had a great introduction by Coval, by Louis, on using renewable electricity, for example, solar, as depicted here in the picture with a solar panel, but of course this could also be other sources of renewable energy, and converting this electrical energy in simple carriers like formic acid, methanol, hydrogen, carbon monoxide. These are all promising, also all different, to gases, to liquid. Formate and methanol are liquid, which makes mass transfer easier. And feed these coming from the chemistry side, the electrochemistry side, at very high efficiencies. You can see here, depends on which numbers you take, but these processes can definitely be more efficient in energy conversion than photosynthesis, and then feed them to microorganisms that make the products that we like, because microbes have been engineered and also are found in nature that can make a broad range of multi-carbon products. So where chemistry is very good in making these one-carbon products from CO2, biology, microorganisms are very good in converting them to multi-carbon products. So that's the whole concept. And just to give another example of how this efficiency-wise and land use-wise actually could have a big impact in the whole green transition that was also nicely introduced by the first speaker. Land use is a major factor, of course, in that. And if you, for example, look at this theoretical calculation, which is actually based on real numbers for solar panel efficiency and also microbial conversion efficiency to protein in this case, so food protein, we can make with what we call electromicrobial route. So, for example, making hydrogen or formic acid from solar energy and feeding that to microbes that make protein. For food, we can feed a lot more people with the same amount of land. So 1 hectare could feed about 500 people in terms of their protein needs per year, was calculated in this nice work. While sugar beet sugar beet feeding to microbes, so that's an intermediate solution. That's basically the more first generation biotechnological production, or the current method of just eating soybeans right away, could feed a lot less people. And of course, if we would compare this to animal protein, it even performs a lot better, but this is based on plant-based photosynthesis. Even the microbial route, electromicrobial route, can potentially be much more efficient in land use. And also we can use non-arable lands. So for example, the solar panels or the wind farms could be on sea or on non-fertile lands. So also realize that this makes, for example, our food system much less dependent on land. I think this is a very important development to look into. But there's a lot of technological and economic challenges for this model as well. And we are mainly looking at technological challenges on the biology side, obviously being microbiologist. So this is once more an overview of the whole concept, combining the best, what we call the best of chemistry and biology. So chemistry is very good, electrochemistry, especially in converting electricity, water, and carbon dioxide, which are in principle truly abundant elements on Earth. Actually, CO2 we even want to get rid of in the atmosphere to some extent. And renewable energy can be scaled up a lot. So these are really scalable renewable resources to make carbon-containing products. Because it's also important to notice that even though we have a big energy transition and we can get rid of a lot of carbon in our energy system, for example, to heat our houses by heat pumps, we cannot get rid of carbon in our carbon-containing products. We cannot make the stable without carbon. We cannot make our food without carbon. And also for some aviation fuels, like you nicely mentioned, this is still a challenge to do this carbonless. So I think this concept is really promising. So we call it electromicrobial production. And so the systems, I won't go into detail on that, but basically as you're combining two systems, an electrochemical system, as we saw in the previous presentation, several nice cells of, has to be integrated with a bioreactor. And this could be done like in the upper graph in one reactor system, which brings some challenges in compatibility, but it could also be an interesting opportunity. But you can also have systems where you have two different reactors, basically, where there is an intermediate fat from one into the other. So this could either be directly feeding, for example, the electrolyte coming out of containing the formic acid, feeding it into a bioreactor, which would avoid some purification steps. But then it means also that the microbes have to stand, for example, the potassium concentrations. So people are looking into that. Another solution is to purify first the formate or the formic acid. That's the last situation, and feed it to the bioreactor. So this is still, I think, ongoing research in many labs, how this could be best connected. Also location-wise, because obviously the first system has to be in one location, while the second and the third system couldn't, in theory, be in different locations. But of course, transport costs are high. So probably co-localizing these systems makes sense at a place where there's a lot of CO2 available, and of course also electrical energy. Good. So then what we are very interested in is also the efficiency of the whole system. So a couple of years ago, we looked into energy efficiency, because we always say it can be potentially more efficient than photosynthesis, and how we currently convert light energy, for example, into carbon chemicals. But what can current state-of-the-art chemical catalysis and biological catalysis do? So just quickly, if you want more details, you can look them up in this paper. But you can see that for almost all of the C1 carriers as well as hydrogen, chemistry is doing pretty well, and about 50% of the energy in electricity, so the input energy here is electricity, can be harnessed into chemical energy carriers. Hydrogen it's the most mature technology, is doing best, close to 80%, but also formic acid, and this is data from a couple of years ago, so maybe you already moved this up a bit, but formic acid is around 50%. CO as well, and for example, methane and methanol can be made in a two-step system, actually not direct electrochemical reduction, but first via hydrogen, also to very high energy efficiency. And also ammonia, by the way, so that's not a carbon molecule, but a nitrogen molecule. So then we looked at the biological side, where our expertise is, and we looked into a wide range of organisms, how they convert this. They convert this via different metabolic routes, metabolic pathways. So that's why there's a lot more bars here, because there's microbes that use, for example, the Calvin cycle, which you find in all plants in the world, but also many microbes, to convert formic acid or methanol into biomass. But there is also organisms that use more efficient pathways, actually. And then we see especially the dark green, the anaerobes, they use a very efficient pathway, so they can convert all these C1 molecules and hydrogen very efficiently. So I think they're very promising, and I will get back to them in the end. The anaerobes, the acetogens, as they're called, because they naturally generate a lot of acetate, but they can also make ethanol. But their main issue is they cannot make a lot more products than ethanol and a few other alcohols. So to make, for example, protein or food, they are not really suitable to do this efficiently. So then we need to move to the aerobes, the light green ones, and they're all way less efficient. So they have conversion efficiencies up to 50% if they have a very efficient assimilation pathway, but several of them are actually down to 20% or so. So for example, the Calvin cycle ones are not so efficient. Good. So if you can add these numbers up, of course, and then overall energy efficiencies depends on what combinations you make. We just highlight some, but they are already very efficient combinations, thinkable like hydrogen or formate or methanol electrochemically, and then using acetogens to make, for example, ethanol. But there's a lot more we want to make, so then we need to go to the aerobes, and there you see the challenge that the overall system efficiency will probably at the moment be close to 20% from electrical energy to the final product, the energy in the final product. So there's a challenge. So, and the challenge is mainly on the biology side, I would say here, the efficiency. So that's what we're looking into. How can we more efficiently convert C1 substrates into products? And another big challenge in the microbiological field is how to make those organisms tolerant to both the substrates, because formic acid is actually toxic if you feed too much to our microbes, and also to the products we want to let them make. So, and then you have a wide range, a huge diversity of microbes that we can find on our planet. And some are better than others, but all have pros and cons. So what we typically do as synthetic biologists is take an organism that at least has some attractive properties, for example, high tolerance to our product or to our substrate, or good genetic tools, and then build genetically by adding DNA and optimizing the other properties. And another property that we actually are highly interested in is the pathway. So as I already said, the Kelvin cycle is, for example, not such an efficient pathway to fix CO2 or formic acid. And we can replace that using genetic engineering by other pathways that take less energy, for example, less ATP, or have faster enzymes in the pathway. So just very briefly on that, because this is a very technical slide, but you don't have to follow all the details. But what we did, this was actually my postdoc work, and we're currently still working on that in my group, is we replaced the Kelvin cycle, which you can see on the right, which takes quite a lot of ATP, in a microorganism by the reductive glycan pathway. So this is another completely different metabolic pathway. It starts at formate, formic acid, and it ends at pyruvate, which is a central metabolite in all living cells that can be converted into biomass or products, other products you like to have. So this pathway could, in theory, yield about 20 to 30% more product from the same amount of formic acid. So basically making this biological conversion more efficient. And what we have shown, for example, in the bacterium Cupiavidis necator, used to be known as Walstonia eutropha, which is nicer to pronounce, but microbes often get renamed in the microbiology field. And this one was already used in industry a couple of decades ago because it makes a lot of bioplastic, polyhydroxybutyrate. You can see the huge blobs inside the cells, and this is basically pure bioplastic. They make up to 80% of their mass in bioplastic. And this is a natural mechanism. These organisms use this kind of as a storage when they are deprived of other nutrients. And so this organism can already grow on formate, but it uses the relatively inefficient Kelvin cycle. And we have genetic tools to basically knock out this Kelvin cycle and replace it by other pathways. And it's also reasonably tolerant to formate. So what we did, and this work was carried on also by the great PhD student Bo Ronsella, who is still working on this in the Max Planck Institute in Marburg, Germany, following up on my postdoc work. And what he has recently shown, which is very exciting results, is that after a lot of optimization of this pathway, so we introduced this reductive glycan pathway, short rGlyP, that we can actually get yield, so biomass in purple, higher than the wild type strain running the Kelvin cycle. So we're now about 10 to 12% higher. So really showing that we can basically beat natural yields by synthetic biology and synthetic pathways. Though if you look at the growth curves on the left, you see that this purple strain improved compared to previous strains that were built in my postdoc, the green one. So we improved, but we're still not as fast as the wild type strain, which doubles every 6 hours. And to really make a good bioprocess, you need to think of both high yield as well as high rate, because otherwise your reactor gets very big to have a certain productivity. So we're still continuing this work. And just to give a short overview, my previous lab and also my current own group are working a lot on this pathway and other pathways in different organisms that are suitable for biotech. For example, E. coli can grow on formate now already. E. coli is a bug that is in our gut. There's also used a lot in biotech that can actually not grow on formate naturally. And now we have a strain that can double every 6 hours on formate. With a reasonable yield as well, but still to be improved. Yeast, which is used a lot in industry, also now people are working on converting this to become a phototroph, Pseudomonas putida, another interesting organism. But none of these works, these are basically all also academic studies that in recent years have shown that we can move synthetic metabolism for C1 assimilation, also for methanol and CO2, by the way, not only for formic acid. But none of these have shown industrially performance levels, let's say. So doubling times, I would say, ideally below 4 hours, and biomass yields, for example, for formate above 4 grams. And that's what we're working on, also in our lab, as well as in collaboration also with industrial partners. And I'm happy to interact with that. With other partners about it. Just to finish my talk, which also as a kind of, I think, an optimistic view on the field, is that electromicrobial production is very promising, but we don't see it upscale yet. But we do see individual elements already upscaled. So we don't see the biology and chemistry units already together at big scale, but we do see, for example, the huge plant that was built, or a pretty big plant on Iceland, where there's a lot of renewable electricity from geothermal energy that makes methanol. You see the plant there in the nice Icelandic landscape that actually makes fully renewable methanol from CO2 and electricity. For hydrogen, this is already done. My neighbor is working on this for formate, so I think we're heading in the right direction. And then on the microbial side, also this is already being upscaled, but the sources there are different. They're not electromicrobial production of C1, but there's basically already in the 70s and 80s, there were big reactors making a feed, animal feed, using microbes growing on methanol. And there is now the very recent plants that are being built, for example, in Ghent and other places in the world by LanzaTech, who are using off gases from steel industry, for example, CO and hydrogen, also C1 carrier, and they are making bioethanol at a large scale with these anaerobes. So this is a plant that was just commissioned in Belgium last year. So I think it's very encouraging to see the upscaling happening, and I think also that what I call synthetic CO2 assimilation pathways and organisms that we are working on bring more future opportunities, especially for aerobic conversion of C1. So with that, I'd like to end my talk and thank a lot of people in my own group and my previous group, the Bar Evan Lab in Potsdam, where a lot of this formate work was done. So with that, I'm ready for the questions.
Thank you very much, Nico. Very interesting talk. So you are proposing, in the meantime, the QR code is visible on screen for people who want to ask questions from the chat. But I will start with my questions first. So you are proposing an electromicrobial approach. So it's a combination of a hybrid chemical and biological process. And of course, as a chemical engineer, I love process intensification. So I'm immediately triggered by this talk. But I also think about upscaling. And you have mentioned nicely that the individual elements are already there at a larger scale. But upscaling at industrial scale, the hybrid process is a completely different challenge. So can you elaborate on that? Which kind of hurdles do you see in upscaling electromicrobial process?
Yeah, so I think for the upscaling, there's a lot of different carrier molecules, for example, that I briefly covered this only, but formic acid brings its own challenges. For example, the electrolyte, which is very salty, is not always ideal for microbial cultivation. So what people have shown, they can either adapt the microbe or the process to that, or maybe partly adapt the electrode process to have, let's say, a compatible carrier, because if you purify, you spend a lot of energy. But if you think about methanol or hydrogen or CO, they're more volatile, so they can easier go out. Basically, the gases CO and hydrogen are, in that sense, easy to go out of the electrolyzer system. But then you have the challenge that we already discussed before of the mass transfer in the bioreactor. So I think it's a really specific case-by-case situation. Yeah. And I think we just need a lot more integration between those fields, because we see the upscaling is there. The gas fermentation by LanzaTech is basically feeding gas, but bringing this together, and I think especially for the formic acid, there is still some challenges, but also opportunities, because it's a liquid feedstock, like is methanol, which makes it for a microbial process easier, because you don't have so much mass transfer issues. So, but yeah, there's some work to be done on these.
Yeah. Okay. Then I hear basically you're saying that having a liquid feedstock helps, but there are still some mass transfer limitations, and there you need to couple with the right expertise to
and compatibility issues, like an electrochemist looks at the at the composition of the electrolyte, and a microbiologist like me looks at the composition of their medium. It's basically two liquid solutions, but there's different wishes. One looks at the optimal performance of the anode, and I look at the optimal performance of the microbe. And we need both in the end. So
yes, definitely. So this is a call for actions to call Nico to all the electrochemists and environmental engineers listening out there. Thanks. And another question. Can you give us some examples of products that would benefit from such an integrated electromicrobial process approach?
Yeah, so I think actually this was already nicely covered also in the previous talks. I really believe if we look at making a big impact, because we can make small products which have a high value already. I mean, we can make pigments, for example, using microalgae or cyanobacteria, but they will not really solve our carbon valorization issue at scale, right? We're talking about gigatons of carbon that go into current chemical industry that we cannot get rid of by just electrifying. We really want to replace the fossils that go into that industry. And the big streams basically are fuels, which we partly need to keep, because probably for heavy duty fuels like aviation, but also shipping fuels, I think that's one. And I think also our food industry, our food and feed industry is very carbon intensive, even though we say always a lot of photosynthesis, but it takes a lot of land, as I showed in the beginning. And I think given especially the promise of microbes having a very high protein content, actually much higher than plants, and very close to the protein content of animal, or actually even higher in some cases, I think the food industry is a very promising one to look at it for this concept as well.
All right, so food and feed and sustainable aviation fuels, or fuels in general.
Yeah.
And is there one specific product that you are already targeting in your research?
So actually, we are looking at the moment a lot at biomass, which is easy for us, and that's food. And actually, for food more than feed, because personally, I believe that if we feed it to animals again, we're again making a very inefficient step in the process. Because so we actually looking at, can we, together with food process engineers, make attractive products from microbes? And that's still, I mean, some people might think, can I eat bacteria or can I eat microbes? But actually we do it a lot. I mean, we are used to making our beers, our wines, our cheeses with microbes, but make them as, let's say, the main ingredient or main protein ingredient, that's still a novel field. And I think using these sustainable C1 and hydrogen feedstocks there, that's where my group is also doing active research together with food specialists, basically at Wageningen University and elsewhere. So that's where I believe we should do a lot of efforts.
Thank you very much. That's very interesting to hear. This leaves us around our 6 minutes before we close. And so I would like to open now panel discussion. We have still a few minutes to have a discussion all together. I do have a very long list of topics I could choose from after such a very interesting presentations. But we need to boil down to one point. And perhaps I would like to focus on purity requirements of feedstock for this biological process. I heard about we need hydrogen, we also need formate, but definitely there are some purity requirements there. Can you, now I'm looking at Tom, I'm looking at Nico, can you elaborate on that? What purity do you need for hydrogen and for formate for your process? Tom first.
It depends a bit on the application. So if you produce a food product, then impurities in your feedstock will end up in your product, which will make it more difficult to sell. You need to guarantee the quality. For the conversion itself, it usually doesn't make a lot of difference. So it will make the biological conversion maybe a bit less efficient, but it's mainly the product that is of concern. So the advantage actually of having such a process, for example, hydrogen production, is that you produce a very clean product, which is gaseous. So if you use gaseous substrates for biomass production, so microbial products, the advantage is that you can easily clean them. So if you have such a relatively expensive production process for your feedstocks, I would definitely also spend some money on quality.
All right, so it depends on the process, of course. Hydrogen is already high quality, but you do need the oxidizer, I guess. You don't want to have oxygen residual in that hydrogen stream.
It depends. So Nico showed some examples of industrially applied biotechnological processes. And I think protein production using hydrogen oxidizing bacteria is moving forward fast. And there you actually feed them with a mixture of hydrogen and oxygen.
Right.
Of course, this comes with safety, but it's again a gas fermentation that you can easily keep clean. And this is a field that is moving fast towards application. So,
thanks. Nico, some comment on the purity requirement?
Yeah, so for formate, I mentioned it a bit already, but I mean, microbes are relatively robust to some small contaminants. I fully agree with Tom that it also depends on the product, but for formate, because it's a liquid feedstock and it often comes out of the electrolyzer as a liquid feedstock already, but dissolved with other salts, we cannot go too high with the salt. So it depends, because at some point, for example, this Cupiavitis nicata that I work with a lot, it's not so salt resistant. So we could also look into more salt resistant bacteria then. We have a lot of flexibility. I mean, the microbial diversity is huge, but we have to make the right choices based on what purity you get. And I think there, the challenge with hydrogen is less. With formic acid, it's higher. But also with methanol, it's probably a bit easier because it's easy to distill to a cleaner level. But yeah, it's not a major. I think it's not a major issue in the field, but you have to take it into account. And also CO2 feedstocks is something we did not discuss a lot, but it's maybe more relevant for you. Where does our CO2 come from? If you feed it directly to the microbial process with hydrogen, you need to feed it to the microbial reactor. Depends where it comes from. If it comes from I think it comes from a power plant where there's also sulfates inside, it might be an issue as well for some microbes, for some not. So yeah, you have to take it into account.
Definitely, yes. But you are also sitting next to a format producer, so do you have any specific questions for Louis in terms of content of format?
Yeah, so I know that some people in this field are working on pure formic acid production, right? That's basically the two methods. They use a gas diffusion electrode, and you focus on the liquid electrode, let's say, and then you produce the salt. So what is your view on these two different? Because for me as a microbial engineer, it's easier if I get it as pure as possible. We can deal with this. But what's your view? Why your process, or will your process outcompete the other process, or do you think they will both develop?
Yeah. So our experience, what we know, like producing formic acid directly in a cell, is more challenging. Because formic acid is acidic, and a lot of metals inside the electrolyzer don't lack so much acidic conditions. Formate it's actually easier to produce, to my opinion, and to what we know. So actually what we're already thinking about is bioconversion processes. We use concentration of 1 molar of formate, and I've been discussing a bit around, like, even for logistic reason, it depends what you do, right? Because if you're a little bit separated, the two processes, the best would be to get a high purity of formate and high concentrated that you can use as a dosing system, for instance.
Right.
That would make sense.
Yeah.
But if you're close by, then you can actually adjust the requirements. You don't have to go you don't have a logistic to take care of. So then you don't have to concentrate your formate or purify it. For purifying, you might have to do that, but so it's all about energy consideration and also cost consideration. And what else to say about the formic acid? We also thought about formic acid would have been easier as far as understood for this kind of process. So what we're also considering is like why not making a kind of a mixture of formate and formic acid? Because every time you purify, you concentrate, you acidify, whatever you do, like if you want very high purity, the cost or the energy requirements always go very high for the last bit of concentration or last bit of purifications. So when we integrate technology, it's always interesting to see like how much you actually need. And if you don't need actually, let's say, like formic acid, not a pure one, but you get 10% of formate, 90% of formic acid, if it's still good, then actually the energy and cost might be much lower compared to 100% of formic acid.
Yeah.
So from our perspective, it's always about like, what are exactly the requirements? How can the range you need to optimize the both system?
Yeah.
Well, it's as you said, once you optimize your system, maybe the other one is getting down in efficiency or cost or up,
Yeah.
So it's always a bit of a
very interesting discussion, but I have to stop it here because the time is running up. But I would like to thank all the speakers again, Nico Claessens, Louis Legrand, Tom Sleutels, and Uffe Thomsen, who just left. thank you again for joining us for this meeting, and thank you all for joining online. And that's all for today. So have a good evening, and see you next time.